好，我们今天很开心来参观 ExoCoBio 的工厂。我上次已经参观过一次了，不过这次来又有更多不同的体验。除了听到我们的 CEO Mr. 赵他有新的一些论述，还有一些新的论文发表以外呢，我看他的硬体设备也改进了很多。对于这个 ExoCoBio 他们家的这个玫瑰提炼出来的外媒体，其实大家有很多疑问。除了上次我跟大家录过的内容以外呢，我今天要来请教他一些大家关于植物性外媒体的一些疑问。Let's welcome Mr. 赵。
Hello， 大家好，我是赵秉成。
中文其实讲得不错哦。I have some question for you because some patient or some client in Taiwan, even the doctors have some questions about the plant-derived exosomes. They say that there are still doubts if the plants have exosomes. Yes or no? And even the plant have exosomes. Is it very effective for human because we are different between animals and the plants? So what's your opinion?
Okay, thank you for your great question. Definitely, all exosomes or extracellular vesicles are released by all kinds of cells. Yeah, so animal, human, plant, even bacteria. They are releasing exosomes or extracellular vesicles to communicate with each other. Maybe 7 years ago, we started from human adipose stem cell exosomes, but just after that, we planned to develop a new type of exosome released by plant stem cells. One big difference between other companies, ExoCoBio is utilizing plant stem cells. Not just plant cells or the whole vegetables, whole plants. They are very different because to develop high quality exosomes, we are using plant stem cell exosomes. And definitely, for example, we started from roses. Why? Because roses are very popular. Every woman loves roses. And then, historically, for maybe several hundred years, people used extracts from roses as a cosmetic ingredient mostly. So our idea was that if we can isolate extracellular vesicles or exosomes from rose stem cells, it would be very different from traditional cosmetic ingredients. And my team was successful in isolating rose stem cell exosomes. And when we characterized those new extracellular vesicles or exosomes, we found that they are very similar to humans, actually. When we analyzed all the contents of rose stem cell exosome by proteins or microRNAs, we found that there are something similar, but something very different also. Because we are different, right? And then one interesting molecule is microRNA LT7B and their family. They are actually very shared by human stem cell exosomes and rose stem cell exosomes.
And also animals.
True, yeah. And we have one more publication on canine adipose stem cell exosome, which contain a lot of microRNA LT7B and their family. So it's quite interesting. And when we treated the rose stem cells or human stem cells, those cells can uptake rose stem cell exosomes, and they are responding. And they released more exosomes after cellular uptake of rose stem cell exosomes. So now we are trying to publish all the data. Internally, we are moving forward to do more scientific research. What molecules or what kind of new molecules are existing in rose stem cell exosomes. So, one more thing I'd like to mention is that there are more than 1,000 scientific publications on exosomes. But unfortunately, plant-related exosomes are just the beginning because we have just less than 500 scientific publications, which is quite a small number of publications when you compare against human adipose stem cell exosome or stem cell exosomes. Definitely, all kinds of cells are releasing exosomes.
In terms of the efficacy, the plant-derived stem cell is inferior to the animal or human-derived?
We found they have very similar effects in terms of in vitro or in vivo assays. But we found also there is some big differences between animals, humans, and rodents. In terms of regulation, as of now, in many countries, we cannot sell human stem cell exosomes because it's changing. We believe that plant exosomes are kind of effective to give some clinical benefits. But the most important thing is that what is the secret of exosomes? So we know all the information, I mean, not all, but the very extensive information on our hands. So our products especially are supposed to give the whole potential of human stem cell exosomes. This is our goal, and everybody in clinic or patient, doctors, they are feeling the clinical effects from our products.
Because some experts in Taiwan is doubtful about the rose stem cell derived exosomes. They think the rose have cell walls. In the literature, the most of the plant-derived exosomes is harvested by the juice, by the juicer, and squeeze the fruit or the leaves. You say you have different methods to obtain the exosomes. You have the rose stem cells derived. What's the technology can release more?
It's a great question. First, we isolate rose stem cells called callus. So when we culture, they can release exosomes into the culture media. It's just almost same as human stem cells. So there is no problem. Most plant exosome companies, they are kind of juice. They crush all the whole plant in one mixer.
Mixer.
They crush everything. So it's a kind of, in terms of human case, it's a conditioned media of stem cells, which contains a lot of unknown, unidentified particle or ingredients. That's why they maybe have no consistent effects. From the beginning, we have a lot of experience on cell culture media, so we don't want any impurity. We wanted to minimize impurities for the best quality and consistent quality. That's why we started from isolating plant stem cells first, and then we culture only stem cells. It's very expensive.
It's your secret technology. That's why you got the patent in US.
Actually, in terms of any plant stem cell exosomes, ExoCoBio is the world first company who achieved a patent in the United States, in Japan, in China, in European countries, actually. And we are the first who registered plant stem cell exosomes as a cosmetic ingredient in the world also.
Another question is, why don't you do the OEM for any demand?
It's a great question. Thank you so much. But, we committed to new technology. Our mission statement is very simple. Original technology, original product, original doctors, original market from the beginning, about 8 years ago. So a lot of big players contacted us so far to have their private brand product, but we rejected every offer. But we are going on our brand.
So thank you so much. It's nice to meet Mr. Zhao. Hope we have more high quality exosome in the future. I hope we can have more application in the future. Thank you so much.
Thank you so much.
Thank you. Bye bye.